The measure of intelligence is the ability to change. Hi guys, Dr. Clark here. Today we're going to discuss the light-dependent reaction, or the light-dependent portion of photosynthesis. Last time we left off, we were talking about an overview of photosynthesis. We were talking about the photosynthesis equation being 6 carbon dioxides plus 12 molecules of water plus photons or light energy is going to yield C6H12O6 or 1 molecule of glucose or fructose plus 6 molecules of water and 6 molecules of oxygen. And remember that oxygen is a waste product in the process of photosynthesis. Okay, so today we're going to dive into a portion of that equation. Okay? Really just the ability for plants to take photons and convert the energy from photons into chemical bond energy, ATP, and utilize the energy or the electrons that's coming out of the system to form an electron carrier, or NADPH. So again, oxygen's a waste product that's going to be removed out of the system, but the rest of this is going to be utilized elsewhere. Before we dive into kind of a step-by-step process of the light-dependent reaction, I'm going to have you guys watch a video that was created about the light-dependent reaction. It dives into Photosystem 2 and Photosystem 1 and the components of these systems. It is semi-technical. We're going to cover a lot of the things that are covered in the video, but some of the things like the specific names of the electron carriers and stuff like that, I don't think it's really relevant for you to learn that. But learning the different systems is important because different products are coming from each system. Okay, the video was created by North Dakota State University. That, for those of you who know anything about me or something about me, that's where I earned my PhD. And so I like to give them as much credit as possible for creating great videos when it comes to these different processes.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: Photosystem 2, cytochrome B6F, Photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, Atp Synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. One photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem II are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem I complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
Okay, so you can see that those photons were coming in and they were exciting, or they were causing resonant energy in the antenna complex. We talked about the antenna complex before, that it consists of chlorophyll molecules and other molecules like accessory pigments like carotenoids, keratins, xenophils. Okay. Now, the video didn't go into all the other accessory pigments, and we're not going to mention too much about them, except for just note that it's not just chlorophyll molecules in the tissue of plant leaves or in the tissue of photosynthetic material, that there are other molecules that can capture the energy from photons and pass it to the reaction center. Okay. So now we're going to dive in piece by piece what's going on, and it's going to be kind of like an overview of what the video just showed us, and with some added pieces, a little bit of added information. First of all, like we said before, that the light-dependent reaction, or the part of the reaction that occurs and needs to occur in the presence of photons, it occurs in the thylakoid membrane, and it's the thylakoid membrane that houses this antenna complex, which consists of proteins and pigment molecules. That's called a photosystem. Okay, there are actually 2 photosystems, and we'll cover each one. Photosystem 2 comes first, and then photosystem 1. Okay, so just an overview, a quick overview. Chlorophyll molecules capture light energy. They pass that light energy from one molecule to another. Again, It doesn't have to be chlorophyll. It can be other pigment molecules, but the pigment molecules capture this energy and pass it from one molecule to another. Eventually, that energy reaches the reaction center, and it excites an electron. The electron is released from the reaction center. The excited electron is picked up by an electron carrier and taken to another portion of the cell membrane, in which the electron is released into another protein. Now, the reaction center, depending on whether we're talking about photosystem 2 or photosystem 1, the reaction center will get a new electron. Okay, it'll get an electron donor. Now, where the electron is coming from depends on which photosystem we're talking about. We're going to start, we'll talk about photosystem 2, and then we'll talk about photosystem 1. They're a little bit different. The products are different, and then what's going into the system is a little bit different also. Okay, so here's that kind of overview. A photon comes in, light energy comes in, reacts with a pigment molecule, chlorophyll, carotenoids, some pigment molecule, okay, excites that molecule or causes resonant energy. Eventually that energy reaches the reaction center. The electron that is housed on that reaction center is excited. It moves to an electron acceptor. That electron acceptor or electron carrier takes that electron elsewhere, and a new electron is donated to that reaction center. Now, in plants and algae, because this is occurring inside chloroplasts, this occurs in two systems, two photosystems. Okay, the first one being photosystem 2. Okay. Photosystem 2 captures that photon just like we've talked about before, excites the electron moves to the electron transport system. Along the electron transport system, a proton gradient is formed and ATP is being produced. Okay, we'll come back to ATP synthase in a little bit. The electron, though, that left the reaction center is replaced by molecular splitting of water, or enzymatic splitting of water, splitting water using an enzyme, using another protein. Okay? We'll come back and talk about this also, because this is kind of an exciting piece of photosynthesis that engineers are starting to pick up and starting to run with. Photosystem 1, photon is picked up again by the antenna complex. Energy is moved, migrates to the reaction center. The electron is excited and moves to another electron transport system. Now that electron that was in that reaction center of photosystem 1, it came from photosystem 2. So when photosystem 2's electron moves through the electron transport system to generate ATP, that electron winds up in the reaction center of photosystem 1. Okay. This reaction center, or this system, photosystem 2, gets a new electron from water. Okay. This system, photosystem 1, gets its electron from photosystem 2. Okay. The product of photosystem 2 is ATP. The product of photosystem 1 is NADPH. NADPH is an electron carrier. The electron that was excited in the reaction center of photosystem 1 is passed to NADPH, and then that NADPH molecule can be used elsewhere inside the cell or inside the thylakoid. Okay? we'll cover each one of these in more detail. I'll show you it in a diagram, and then we'll keep covering it. I know this is going to be new for a majority of you, that you've probably never heard of this before. So we'll cover it multiple times. All right, so let's look at the two photosystems. The first photosystem is called Photosystem 2. Sometimes it's also called P680, okay, which refers to the reaction center. All right, so Photosystem 2 has a reaction center called the P680 site. It's holding an electron. That electron came from the splitting of water, the enzymatic splitting of water. Now, the reason why I bring up that water splitting is it's important for modern day, because as we're progressing towards moving away from fossil fuels and we're interested in renewable energy, well, this might be an option. There are quite a few individuals out there trying to make hydro engines, so cars that are ran on water, motors that are ran on water. And one way to get at that is to do what plants have been doing for 1 billion years, and that is split your water using an enzyme. If you can use an enzyme, a protein, to split the water, the electrons will be released, the energy will be released, oxygen will be released as a waste product. So that would be what's coming out of your engine. And then you can deal with hydrogen, with protons. Right now, how to deal with the hydrogens, the excess hydrogens, can be problematic if they're allowed to form like hydrogen gas, etc. So it's not perfect. We haven't figured it all out yet, but there are a lot of engineers that are working with the ability to do exactly what plants have been doing for the last 1 billion years, and that is get their energy from splitting water. All right, so that electron is redonated to the P680 site. The electron is excited. Okay? It's carried to the electron transport system. The electron moves through a protein, and that protein is often called a proton pump. So it's pumping protons into the thylakoid space. So you get a gradient. You're forming a bunch of protons on one side of the membrane. Okay? The other side of the membrane has very little protons. So these protons want to go back across. The only way these protons can go back across is if they go across through another protein called ATP synthase. We'll come back and look at this. The electron's donated to P700 site. It's excited. It goes to the electron transport system. Now that electron is picked up by NADP to form NADPH. Okay. All right, so let's look at this again. Plants produce ATP and NADPH. Those are the products, at least those are the products from the light-dependent reaction, is ATP and NADPH. And we say that it's produced in a non-cyclic, so in a linear form, okay? And it's produced via non-cyclic photophosphorylation. Now, the photo part refers to photons, right? So light. The phosphorylation refers to the addition of a phosphate group to this molecule. So if you remember from the video, ADP, or adenosine diphosphate, plus a phosphate is going to form ATP. Well, that formation of ATP is called phosphorylation. When you utilize light, when you utilize photons to do it, it's called photophosphorylation. Okay, so what do you do with those extra electrons? So you form ATP, but you still have an electron that's moving through the electron transport systems, well, those electrons are picked up by NADP, okay, to form NADPH, which is an electron carrier, and it can carry it to another portion of the chloroplast in order to drive work elsewhere. So the general products of the light-dependent reaction, there's two real products that come from it, and that's ATP and NADPH. Now there's a waste product that comes from the splitting of water, and that's oxygen. Right? But the products that the plant is generating is ATP and NADPH. Let's look at how this is done. Okay. So again, now the diagram is showing you the thylakoid membrane. So remember, inside the chloroplast organelles, is thylakoids. Thylakoids, okay, are those little stacks, and if you stack a bunch of them together, they're called grana. But thylakoid is that little sac-like structure. Has a membrane, lipid bilayer. Looks similar to this. Okay. That thylakoid membrane has proteins bound inside that membrane. And those proteins, some of them are antenna complexes, which allows for them to capture photons. Okay, so they would have molecules like chlorophyll, carotenoids, keratin, xenophils, pigment molecules that can capture light energy. Okay. Also, they're going to house an electron in the reaction center. Water is going to be split so that electron can be donated to that reaction center. As a waste product, oxygen is formed, and then you got a bunch of protons or hydrogen ions that occur in the thylakoid space. Now, we talked about this before. Okay? Electron excites or a photon comes in, excites the electron. The electron moves to a carrier, is passed to another protein. This protein is sometimes referred to as a proton pump because it's pumping hydrogen ions across the membrane, because hydrogen ions can't move through this membrane because they have a positive charge. So it's pumping them across and creating a proton gradient, a bunch of protons on one side of the membrane and very few on the other side of the membrane. That electron is then moved to another carrier, excited again in photosystem 1, okay, and then moved to another protein. In this case, this protein is forming NADPH because the electron is being picked up by NADP and a proton is being added to it to form NADPH. Now, I told you photosystem 2, the product of photosystem 2 is ATP. So in this membrane, often all over in this membrane, you're going to have ATP synthase. You're going to have a specific protein that allows for these protons, these hydrogen ions, to move back across the membrane. In doing so, it generates ATP. Okay, so we're going to look at that next. So, the result of pumping protons across, you get a large concentration of protons on one side of the membrane and very few on the other side of the membrane. Like I said before, the thylakoid membrane does not allow protons to move across that membrane. They have a charge. Charged or ion molecules cannot move across membranes, typically. They can move across the membrane, but they have to go through a protein channel. The protein channel that these protons or hydrogen ions have to go through is the protein known as ATP synthase. When you see ASE at the end of a molecule, it often refers to an enzyme. So this is the enzyme that generates ATP. Protons, again, we kind of touched on this before, that material in living systems is often trying to reach equilibrium. So if you push a bunch of stuff on one side of the membrane and you have very little on the other side of the membrane, that material wants to go back across and reach an equilibrium between the two sides. Okay? This process of moving material across the membrane is called osmosis or diffusion. Okay? It depends on what the material is that's moving across the membrane. Okay? So moving material back and forth is very common. It's a common practice, and living systems are trying to reach homeostasis or equilibrium as often as possible. This gradient, or by pushing a bunch of protons on one side and then having them come back across, generates energy. And that energy is enough energy to take ADP, or adenosine diphosphate, phosphorylate it, so add another phosphate group, to form ATP. The process is called chemiosmosis. So I just told you that movement of material across the membrane is called osmosis. In this case, you're forming chemical bonds by utilizing the movement of material across the membrane. The only way that that material can get across that membrane is through a protein channel. That protein channel is called ATP synthase. And so you're through mutations in your body, well, in this case, in plant tissues, eventually this process evolved, and now we have a process that utilizes proton gradients to drive work. So it's becoming, a fairly streamlined or efficient way to generate energy. We'll look at ways where it breaks down, though, in a little bit. All right, so going through that chemiosmosis or ATP synthase. Again, photon comes in, photosystem 2, excites the electron. Okay? The electron moves, it moves through a protein. That protein becomes a proton pump, pumping hydrogen ions across the membrane. Okay, so again, hydrogen ion and proton in this case is used interchangeably. It means the same thing. Okay, so those protons or hydrogen ions are moved across into the thylakoid space, creating a gradient, okay, a proton gradient. These protons need to get back across to reach equilibrium. They do through ATP synthase, and ATP is formed from ADP and inorganic phosphate, and the energy utilized from moving things through this protein. Okay? Next time we're going to talk about the Calvin cycle. So how do plants utilize the ATP and the NADPH that's generated in the light-dependent reaction to drive the work that's needed in the light-independent reaction? It's these three gentlemen, okay, Calvin, Benson, and Bassam, that came up with that process known as the Calvin cycle. Next time.